Hello and welcome to our second video in our Chapter 42 video lecture series where we are going to talk about adaptive immune response. And our learning objectives are to explain adaptive immunity, compare and contrast adaptive and innate immunity, describe cell-mediated immune response and humoral immune response, and describe immune tolerance. So first, a bit about adaptive or acquired immunity. This type of immunity will take days or even weeks to develop. It is going to take much longer than our innate immune system, which is kind of already set. It is there, especially our physical barriers, right? This immune system is more specific to pathogens because it is tailor-made, and we're able to keep an immunological memory, which gives us a long-term protection if we were to become infected with the same pathogen again. And this occurs after exposure of a specific antigen takes place, or it could be due to a vaccination in which we are giving maybe a weakened version or a killed version of that pathogen. We can divide the adaptive or acquired immune system into two subcategories: cell-mediated immune response, which you could see listed over here, and this is really going to be carried out by our T cells. You could see our two different types here, the helper and the cytotoxic T cells, to combat pathogens. And our other one is going to be our humoral immune response. This is also called our antibody-mediated response, and it's largely controlled by our B cells. And our B cells will proliferate into something known as plasma cells, which then are going to synthesize and secrete antibodies. Now, before we go any further, let's talk a bit about the types of T cells, because we are going to be using this terminology. We need to know what they are. So first we have our cytotoxic T cells. These are also known as our killer T cells or CD8 cells, and their job is to destroy virus-infected cells in our cell-mediated immune response. They're also going to play a big role in combating cancer cells. So I put the little axe symbol here to symbolize that they are going to have a direct attack of these cells. The other type we have are our helper T cells. These are also known as our CD4 cells, and they play a part in activating both our antibody and cell-mediated immune responses. So it's really the one that is going to sound the alarm to our immune systems in our adaptive immune system. And then lastly, we have our suppressor or regulatory T cells, and these are going to deactivate our T cells and B cells when needed. So you might be thinking, why are we deactivating it? Don't we want to activate them so they combat the pathogen? Yes, but we also need someone, an immune cell, to stop the attack when the pathogen is already gone from our system. So this really stops our immune system from being too intense. So we put the little stop sign here, and if our regulatory T cells are not functioning properly, this could lead to an autoimmune disease. We're going to talk more about that in section 4. Next, let's talk about our antigen presenting cells, or what we call APCs. First, let's redefine what the term antigen means. This could be a foreign or non-self macromolecule that reacts with cells of our immune system. Now, we have mentioned our self antigens before. Remember that our own self antigens should not produce an immune response. We also may be exposed to harmless foreign antigens such as food proteins, pollens, or dust components that should not trigger an immune response. Now, we know for some people they do trigger an allergic response. Again, we'll talk about this in section 4. Now, back to our APCs. These are specific immune cells that are able to detect pathogens, engulf them, and also, by way of what we're going to soon learn about an MHC molecule, inform our adaptive immune cells by presenting that antigen that it took off of the pathogen to the immune cells. So we have three main antigen presenting cells. We've got our dendritic cells. that we are going to find mostly within our skin as Langerhans cells, and also within areas like the lining of our nose, our lungs, stomach, intestines. We'll also find them in other places like our lymphatic structures, but more on them later. Our macrophages will also be an APC, and our B cells, as long as they haven't been activated and started the differentiation process. So next, let's talk about the MHC molecules. This is going to be a protein structure in which we are able to break off a piece of the pathogen, also the antigen on there, that is going to be displayed on what we call an MHC, a major histocompatibility complex. Now we do have two different classes, Class 1 and Class 2. Class 1 is found on all of our nucleated cells. So you'll find it on a liver cell, a gastric cell, a muscle cell. Will we find it on a red blood cell? The answer would be no, because a red blood cell does not have a nucleus. Now, our MHC Class 2 molecules are only found on antigen-presenting cells. And these antigen-presenting cells will also have Class 1 MHC molecules because they are nucleated. So you could see one of those APCs over here. We have our MHC Class 1 molecule with an antigen on it, and MHC Class 2 with an antigen on it as well. Don't worry so much about what's going on here. We're going to talk more about that later on. But before we get into more of the MHC reaction with immune cells, let's talk about how we display it, the pathogen itself on there. So when we have an APC, we find that bacterium or foreign pathogen that is ingested through endocytosis. From there, we are going to create a phagocytic vesicle that will then fuse together with lysosomes. If you remember, the lysosomes have digestive enzymes within them. So when we combine it, we're going to call it a phagolysosome, and we will start to break down the pathogen's components into teeny tiny fragments. Then we will take an antigen and place it on an MHC Class 1 or MHC Class 2 molecule and exocytose it and display it on the cell surface of our APC. At that point, our helper cell is going to be one of the main immune cells that comes over and responds to the antigen presenting cell. That is kind of what we're seeing over here. We would have this reaction over here. We're going to talk about what CD4 and TCR, the T cell receptor, is, but here you can see the MHC Class 2 molecule binding with those structures, and it is going to help activate this cell so that it can trigger an immune response. Now let's talk a bit more about our T and B lymphocytes. Our lymphocytes that are circulating are mostly our T cells, about 80 to 90%, and they are going to respond to the APCs, as we said before, and indirectly induce an immune response by releasing cytokines. They'll also stimulate our B cells to prepare their own response. We know that's a humoral response where we're going to produce antibodies. We also detect the APC signals and directly kill the infected cells. These, of course, would be our cytotoxic T cells. And then suppressing inappropriate immune reactions, those would be our suppressor T cells. And the other 10 to 20% of our lymphocytes are our B cells. Now that recognition or binding of specific antigens are going to happen through a complementary receptor. And once they bind, we get activation and we will self-amplify, meaning we're going to copy that immune cell and make it tailor-made to that specific foreign antigen. When we get activated, it is because we have these smaller components on an antigen known as an epitope. And because they're so small, we also call them antigenic determinants. This is going to be important later on, so keep this in mind. Now we go through a selective process for our T cells. We know that our T cells originate in the red bone marrow, and then they're going to travel to the thymus in order to mature. So through thymic selection, we'll eliminate about 98% of those T cells produced. And we then will have 2% that survives that selection in the thymus. So when it comes to what we call our naive T cells, we see that they will have both the CD4 and CD8 proteins on them. Now at this point, we are ready to differentiate. So our helper T cells will lose the CD8 protein and keep their CD4 protein. This is why they're also called CD4 cells. Whereas our cytotoxic or killer T cells will lose the CD4 protein and keep their CD8 protein, which is why we call them CD8 cells. And our molecules regulate how a T cell will interact with and respond to an APC. So how do we activate these T cells? First, let's talk about our naive CD4 cells, which is pictured over here. We see our antigen presenting cell that has its MHC2 molecule out with the epitope on there. And we're going to see that there's contact between the CD4 protein and the T cell receptor. When that match is made, they're both going to release cytokines, and the T cell starts to clone itself that has the exact match for that epitope that was on the APC. And then once it clones itself, all of those cells are going to secrete specific kinds of cytokines that can activate the B cell so that it can go off and produce antibodies, and the CD8 cells are killer T cells so it can go and kill cells directly. We'll talk about that mechanism in a few slides. Now with our naive CD8 cells, this is going to engage with our MHC1 molecule with the epitope on it, and when it is a match, that killer T cell is going to produce two types of substances. We have our granzymes and perforins. Essentially, the perforins create little openings within the infected cell, and the granzymes go in and cause the cell to apoptose. And then the cytotoxic T cell can also emit some cytokines so that it can amplify the immune response. Now, both of these types of T cells are binding to T cell receptors and the co-receptor. In this case, that co-receptor was CD8. In this case, it was CD4. Let's talk more about those T cell receptors. We'll see that each one will consist of two polypeptide chains that will span the T cell membrane, the plasma membrane that surrounds the cell, and it's bridged by a disulfide bond right over here. We have this region in purple that remains constant, and this is its intracellular domain that's involved with intracellular signaling, whereas the variable region is going to differ within each TCR. And this creates TCR diversity, which is achieved by mutation and a recombination of genes, which happens at random. So we literally get a ton of different combinations that can be prepared to bind any assortment of epitopes or antigens. Now, a bit more on our helper T cells. They're important for extracellular infections, such as the ones caused by bacteria, helminths, and protozoa. And they recognize specific antigens displayed on those MHC class II complexes of our APCs. And we do have two subdivisions of these helper T cells. We have Th1 cells. Their job is to secrete the cytokines to enhance activities of our macrophage and cytotoxic T cells and some other T cells. So they're associated with inflammation, and also they work more toward intracellular invaders. So think more of virus-infected cells, for instance, or maybe certain bacteria like Mycobacterium tuberculosis that enters our macrophages and essentially multiplies within them. The Th2 cells are more for stimulating our naive B cells so that they can proliferate and create antibodies specific to that antigen. So that's more aimed toward our intracellular invaders, our extracellular invaders. A bit more on our B lymphocytes. When they get stimulated by our Th2 pathway, right? We just said those are the specific ones that stimulate our B cells. The naive B cells become activated and start to differentiate. So they become plasma cells, which then are going to synthesize and secrete antibodies. Whereas on the other side, they are going to create some memory B cells so they can remember this antigen, and it doesn't really do much during this initial infection, but it'll keep record, and that way when we are exposed to this antigen again, it can quickly differentiate into these plasma cells and create these antibodies. Now we, of course, have some B cell receptors. They look a bit different than our T cell receptors. When our B cell is naive, they are going to be coded in 1,000 BCRs. And notice how these are looking like antibodies if you have ever seen them. And our antibodies are also called Ig or immunoglobulins. And so when they're naive like this, they're really just functioning as APCs. They have their constant region here that's embedded within the cell plasma, and then their variable region is going to be similar to the TCR in that we are going to have mutations and recombinations to create hundreds of millions of different BCRs to give extensive diversity. Now they're going to bind and engulf a foreign antigen through their BCRs and then be able to display those processed antigens on their MHC2 molecules and present them to our Th2 cells. So how do we activate them exactly? Here we see our naive lymphocyte. We got that antigen, so we got to process it and then place it on our MHC2 molecules. Now you could see over here it's being presented to this helper T cell. We're going to bind to its co-receptor of CD4 and its MHC class 2 molecule here with its TCR. That activates the helper T cell. And now we're going to produce cytokines. You can see the cytokine here is IL-4 that will cause this B cell to differentiate and proliferate. And so it creates copies of itself, and that is called clonal selection. And now we're able to create these plasma cells that will synthesize and secrete the antibodies specific to that initial antigen. Like I said, we also create those memory B cells. All right, next up are our cytotoxic or killer T lymphocytes or CTLs. They're really important, once again, in protecting us from viral infections and also cancer cells. When our APCs phagocytize a pathogen, you could see that taking place over here with this macrophage, they're going to display it on their MHC1 molecule, and then our killer T cell is going to bind to it and become activated. They then will proliferate through clonal expansion or clonal selection and go in search of cells that have that same antigen displayed on their MHC molecule and destroy them. Now, we mentioned already how our cytotoxic T cell is going to secrete perforins and granzymes. So I wanted to show you an illustration here of those perforins coming in, creating pores that then allow granzymes to move in, and that induces apoptosis of the infected cell. Now note that our plasma cells and our cytotoxic lymphocytes are collectively called effector cells. They are going to do the work of essentially the immune response. They're directly working on our infected cells, whereas our memory cells do not do that. They are not effector cells. So if our, let's say, NK cells, which act really similarly to our cytotoxic T cells, can't identify the missing self pattern of a downregulated MHC1 molecule, then our cytotoxic T cell can identify it and release its perforins and granzymes. And if we had the opposite take place where our cytotoxic T cell isn't able to detect the antigen MHC molecule combination, then the NK cell can come in and destroy it in itself. So this way they kind of work together in order to kill off the infected cells. And remember that when those immune cells come into contact with any of the MHC1 molecules that is displaying a self-antigen, it should not destroy it. We only want to destroy it if we see that foreign antigen on it. Now let's talk a little bit about mucosal surfaces. We have mucosal immunity within our mucosa-associated lymphoid tissue, also known as MALT. So this almost is its own immune system that works independently from our systemic immune system. And we have its own innate and adaptive components. It'll create an immune barrier from pathogens because we are going to have contact to the external environment. So we find them in areas such as like our tonsils, our adenoids, which are found in our nasopharynx, our appendix within the large intestines, or coming off of the large intestines rather, or our mesenteric lymph nodes, meaning the lymph nodes that are surrounding our intestines, and around the lining of our GI tract, respiratory tract, and urogenital tracts. So essentially, all of these tissues are a collection of lymphatic tissue that is combining with the epithelial tissue that lines the mucosa within our body. And in here we have these M cells that are absorptive epithelial cells, and they're the ones that are taking up the foreign particles and deliver them over to our APCs. Remember, that's going to include B cells, macrophages, or dendritic cells. And then from there, our APCs, which tend to be mostly dendritic cells in MALT, will travel through the lymph into a lymph node, and then that eventually can take it over to our circulatory system. Another important thing we need to do is build immune tolerance. This is our ability to prevent an unnecessarily harmful immune response to a detected foreign substance that isn't really known to cause a disease. So think like a food protein such as peanuts. And this is going to be crucial for maintaining our mucosal homeostasis, because our mucosal membranes come into contact with a lot of different foreign substances. I mentioned food proteins, but we also have pollen and dust and all of that as well. And so this can really be brought about through our specialized APCs in areas like our liver, lymph nodes, small intestines, and our lung, where we present it with harmless antigens, and our regulatory T cells are going to work to suppress local inflammation and inhibit secretion of stimulatory immune factors. Now, if they aren't able to do this, then it can trigger an autoimmune response or a hypersensitivity, which we're going to get into in section 4. Another function of our regulatory T cells, of course, is to stop the immune processes to a harmful pathogen once our infection has been cleared. And I've mentioned the immunological memory beforehand, but now let's talk about it in the sense of both our memory B and memory T cells working together. So when we first get infected with a pathogen, that's called our primary response. We're going to have our plasma cells develop and secrete antibodies, and the differentiated T cells will start to increase. And then what happens is they start to plateau over time. So this graph over here is showing us healthy, then we get infected with the pathogen, and we're going to see that it takes some time for that primary response to take place, for us to build up and create those antibodies. So this is taking, days over weeks here for us to develop this primary response. And then we created memory cells here, B and T memory cells, from this primary immune response, right? So remember that those were not effector cells. They're just staying within circulation, and they can circulate for years even. And eventually, after several decades, they can die off. But if we were to come into contact with that same pathogen, then they can immediately become effector cells and produce a secondary response. And you could see that it is way more rapid. It happens very quickly. And we are going to be able to output tens to hundreds fold of greater antibody amounts than we produce within our primary response. So this is really why our vaccines work, is because we give the, let's say, wanted poster to our immune cells and generate a mild primary immune response, so that when we become infected with that actual pathogen, we're able to give this explosive immune response to it and fight it off. Now, sometimes those memory cells are circulating for so long, like we said, that they die off. And this is why for certain vaccines, we need boosters. For instance, our tetanus booster is needed every 10 years because our memory cells will only live out that long. And then let's talk about our mucosal immune memory. They are going to also have memory B and T cells in there. And if we're reinfected by that same pathogen, we get that same pronounced immune response at the mucosal site. Now let's talk about the primary centers of our immune system. Our cells of the immune system can travel between distinct lymphatic and blood circulatory systems through extravasation, essentially meaning that we are able to shift between the blood vessels here into the tissues and then into our lymphatic system. So if we think back to what we learned in our blood system, our immune cells are going to originate through hematopoietic stem cells in our bone marrow. And then we have cytokines that stimulate the stem cells to differentiate them into immune cells. Our B cells are going to stay within the bone marrow to mature, whereas our T cells will travel over to the thymus in order to mature. Now notice how we have two different types of selections that take place here. Positive selection is when we're going to have survival that is dependent on our ability of our T cells to be able to bind to MHC molecules. So if they are able to bind, they survive. If not, they're going to be destroyed through apoptosis. Whereas negative selection is really checking to see if they're going to be able to recognize our self-antigens. So if they are able to recognize them and bind to them, we want to destroy them. Otherwise, we would have an autoimmune disease. If they don't bind and essentially would trigger an immune response, then we want to allow them to survive. And lastly, let's talk about what happens to our mature B and T cells after they go through that positive and negative selection. When they're mature, they're going to circulate and travel over to secondary lymphoid structures, meaning that they're traveling to our different lymph nodes, the spleen, and even maybe the tonsils. And so that lymph that's traveling through our lymphatic system is going to gather antigens as it drains the tissues. And as that lymph travels through, it's going to come into contact with our lymph nodes that will filter that lymph and have some APCs, antigen presenting cells within them that could capture those antigens and process them. And there's lymphocytes that we'll find within here that we could present them to. Now, let's say it surpasses those lymph nodes, and in our lymphatic system, we travel all the way back up here, and we put those antigens within our circulatory system. It could either be on its own, or let's say it came into contact with an APC in the lymph node, but we couldn't find any immune cells there. So we're going to circulate now into our blood, and from the blood, eventually it is going to travel over to our spleen. Our spleen could be found in this upper left quadrant of the abdomen, where we're going to house our B and T cells, some macrophages, and dendritic cells and NK cells. So if we have an APC that has an antigen there, it can communicate it with the lymphocytes in this area. And then if our B cells are activated, we can get those antibodies synthesized and secreted from the plasma cells in the spleen.